Today I would like to discuss the generation of visual action potentials in differently lit environments. To start off with a little background, this is the structure of cells in retinal layers of the eye. Firstly, the rod cell is a light-absorbing photoreceptor cell in the retina. They are used in monochromatic vision and help us to see in low-light environments. They are also crucial in the generation of peripheral vision. The bipolar cell is a retinal interneuron that provides a pathway from the rod and cone cells to the retinal ganglion cells. Retinal ganglion cells are cells that connect the rod and bipolar cell to the deeper vision processing centers within the brain. In this first case, where there is no light to be received, rhodopsin in the rod cell absorbs no light. Rod cells are depolarized in this low-light environment. Taking a closer look into the rod cell in this case, depolarization in this case means that positively charged sodium and calcium ions are permitted into the cell, which decreases the membrane potential. This depolarization triggers an action potential which causes the neurotransmitter glutamate to be released by the rod cell into the synapse between the rod cell and the bipolar cell, which hyperpolarizes and inhibits the bipolar cell. Since the bipolar cell is inhibited, there is no synaptic activity between the bipolar cell and the ganglion cell, and therefore there can be no signal from the retinal ganglion cell to the optic nerve fiber. Baseline firing occurs, as shown in the graph to the right. In this case, light is available, and rhodopsin in the rod cell absorbs light. As little as 1 photon is required to elicit an effect. The rod cells are hyperpolarized by this. Taking a closer look into the rod cell, hyperpolarization means that positively charged sodium and calcium ions stop influxing into the rod cell and create a large membrane potential. This hyperpolarization causes glutamate secretion to stop at the synapse between the rod cell and the bipolar cell. Since the bipolar cell is no longer inhibited by glutamate, the bipolar cells are depolarized and start releasing neurotransmitters into the synapse between the bipolar cell and the retinal ganglion cell. Because of this, the retinal ganglion cell is highly stimulated and sends out a quickly spiking firing pattern, as shown on the right. The axons of the retinal ganglion cells carry certain spiking patterns from this retinal structure in the eye and form the optic nerve. Because of this, the optic nerve fiber can facilitate a signal to deeper visual processing sites within the brain.